Selamat siang Pak Wayan. Pak Wayan Mudianta, selamat siang. Masih mute Pak.
Selamat siang Pak Pras.
Ya,
Selamat
silakan.
siang Mbak Resi, teman-teman semua. Luar biasa bisa bersua kembali hari ini. Oke.
Selamat siang Pak Wayan, Pak Pras, dan rekan-rekan.
Ya.
Good afternoon Ernesto.
Hi Wayan, good morning everyone.
Okay, so
good afternoon, sorry, good afternoon for you.
yes, morning from Italy and afternoon from Bali. Okay, I'm glad to see you again. So how are you doing?
Great.
Okay. Yep. All right, so we might need to wait for a couple of seconds. Just wait for the audience to join with us. So at the moment we have 30 participants in the room. So, okay. All right, just make sure that Prof. Ocky and Pak Made Gunaja are also in the room. So Resi, can you confirm with me if Prof. Ocky already joining with us today, the keynote speakers?
Yes, sure, Pak Wayan.
Okay.
I see Pak Gunaja maybe already here.
Okay.
Brida Prof. Bali ya.
Oke. Selamat siang Pak Made Gunaja. Halo, selamat siang. Oke, kita tunggu beberapa saat. Prof. Ocky, selamat siang. Oke. Oke, untuk panitia coba dikonfirmasi dulu ke Prof. Ocky dan Pak Made Gunaja sebelum kita mulai untuk pembukaannya. Teman-teman yang lain, peserta, mohon kesabarannya untuk menunggu beberapa saat sebelum kita mulai. Oke. Oke.
Ya, Pak Gunaja sudah terlihat di layar, Pak Wayan.
Oke, waduh. Selamat siang, Pak Made.
Selamat siang, Pak.
Nggih, apa kabar
Wayan.
Pak Made? Nggih, senang
sehat,
bisa
sehat.
berjumpa kembali ini. Nggih.
Selamat siang Pak Made.
Selamat siang, Pak Pras.
Udah lama saya tidak ke kantor lagi nih. Iya. Masih sibuk Pak Made, Pak Gunaja?
Iya, ada nih, kita merancang kegiatan nih tadi karena masih dalam rangka untuk pembentukan BRIDA ini.
Oh.
Pematangan tadi ikut bagaimana implementasi kebijakan pusat berkaitan dengan BRIDA ini.
Ganti nama ya Pak Made ya, dari BARI menjadi BRIDA sekarang ya.
Kita siap support Pak.
Ya, aja ini Pak Ayan, artinya akronim kita ganti. Akronim kita kan dulu BARI, akronim kita sekarang namanya tetap Badan Riset dan Inovasi Daerah, akronimnya jadi BRIDA bukan BARI gitu.
Iya. Oke. Nggih. Tempatnya masih sama ya Pak ya? Kantornya masih sama. Nggih. Oke. So, Resi dan panitia, Prof. Ocky gimana? Udah konfirmasi beliau bisa join di room?
Prof. Ocky sudah konfirmasi Pak Wayan, mungkin nanti menyusul.
Oke. Alright, kalau begitu bagaimana kalau kita mulai saja? Ini kita sudah ada 36 peserta yang join di room ini. Di jam saya jam 2:08.
Mungkin kita bisa putar video lalu mulai Pak Wayan.
Oh, silakan. Oke.
With over 17,000 islands, Indonesia is often called the world's largest archipelago. All these islands cover only about 30% of the whole country. The other 70% is water. This in mind, it is easy to see how immensely diverse aquatic ecosystems develop here. Indonesia is a country with great potential in marine mega biodiversity. Qualified human resources are needed to research and get a better understanding of this extraordinary potential. This will have positive impacts on many levels for future generations. Proper infrastructure and developing knowledge are the base to be able to go deeper into modern marine research. MERO, which stands for Marine Education and Research Organization, is supporting this specific line of research. MERO was founded in 2020. This institution is located close to the sea on the famous Raya Secrets muck dive site in the Tulamben area. The Tulamben area is home to a wide range of various underwater habitats, including a shipwreck, coral reefs, sand slopes, rocky areas, as well as muddy inflows from rivers. MERO students have the possibility to explore biodiversity directly by diving here. MERO's laboratory is well equipped with the latest in technology, where not only marine research is done by a qualified team, but a special aim is to offer the possibility to students from Indonesia to learn and share on a national and international level. Already collaborations are active with many national and international scientists and students. MERO's laboratory provides research equipment for microbiology, morphology, chemistry, DNA processing, as well as a scanning electron microscope. It offers students and researchers accommodations like dormitories and private rooms. There is a spacious classroom for courses and seminars. More buildings like an additional laboratory, a restaurant, and a showroom are planned. MERO was built in the idea of sustainability and eco-friendliness in mind. Electricity is provided by solar panels. Wastewater gardens take care of the sewage. Garbage is separated, and the organic parts directly composted on the grounds. Being in a rather rural area, MERO also supports the local community with education in regard to garbage management, and there is a Sunday class for the children from the village. The very heart of MERO is certainly marine education and research in general, and the research of nudibranchs in specific in collaboration with scientists from all over the world while offering students to take part in this process and knowledge. MERO is ready to welcome interested students and researchers.
Oke, Resi, apakah kita bisa mulai?
Yes, Pak Wayan.
Oke, thank you. Terima kasih. Videonya sudah ditonton tadi, filmnya udah selesai segitu, karena durasinya pendek, memang iklan. Ya, oke, alright. So, Om Swastiastu, Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera untuk kita semua. So, welcome to the MERO Foundation webinar series in this year. So the topic is about marine invertebrate-associated microorganisms. I'm Wayan Mudianta. I'm the facilitator in today's seminar. I'm also a member of MERO Foundation, and then before we start, I would like to acknowledge to Dr. Ernesto Mollo and friends, and also the participant who might not speaking Indonesian. This seminar will be held in bilingual mode. So the first part of the seminar will be held in Indonesian, including the welcoming speech from the foundation, from the founder, and also from Pak Made. But the rest of the conference, the rest of the seminar will be held in full English. However, if you might need assistance in language translation, I would be happy to help you. Okay, so for Ernesto and friends, thank you for your understanding. Oke, pertama izinkan saya untuk menyapa undangan yang luar biasa, Pak Made Gunaje. Kemudian ada 2 keynote speaker kita pada hari ini, dan juga teman-teman dosen, Bapak Ibu, mahasiswa dan peneliti yang sudah terdaftar atau mendaftar di seminar kita kali ini. Di catatan kami ada 80 orang, sampai saat ini baru 40 orang yang join, kita tunggu beberapa saat lagi. Oke, yang terhormat Bapak Insinyur Imade Gunaje MSI, beliau adalah Kepala Badan Riset Inovasi Daerah Bali. Ini suatu penghargaan yang luar biasa bagi kami, karena di tengah kesibukan beliau sempat meluangkan waktu untuk untuk hadir dan memberikan sambutan pada acara ini. Perlu saya sampaikan bahwa ini adalah kehadiran kedua kalinya beliau pada acara seminar yang diadakan oleh MERO. Saya masih ingat tahun 2020 ketika MERO mulai berdiri, beliau juga hadir dan memberikan materi pada suatu seminar. Nah, hal ini membuktikan konsistensi beliau untuk terus mendukung perkembangan riset di Bali dan juga di Indonesia secara umum. Jadi kami sampaikan apresiasi yang setinggi-tingginya kepada Pak Made. Selanjutnya kita ada 2 keynote speakers dalam seminar ini, keynote speaker yang memang sudah sangat berpengalaman dan ahli di bidang mereka masing-masing. Pertama kita punya Dr. Ernesto Mollo, ya, dari namanya ini beliau Italia ini. Jadi dari Institute of Biomolecular Chemistry, CNR Italia. Jadi kayak lembaga riset nasionalnya Italia. Nah, beliau adalah salah satu pakar di bidang chemical ecology terbaik di dunia. Karena sudah lama banget dan produktif hasil-hasil riset beliau. Nah, kemudian yang keynote speaker yang kedua adalah Profesor Ocky Karna Radjasa. Jadi Prof. Ocky Karna Radjasa adalah beliau adalah ketua organisasi riset di bidang kebumian, di BRIN, Badan Riset Inovasi Nasional. Jadi pada seminar kali ini kita mempertemukan 2 tokoh badan riset ini. Jadi ada di nasional, ada di daerah. Prof. Ocky, ya, sering dipanggil Prof. Ocky, beliau ahli di bidang marine microbial symbiont, riset-riset beliau sangat mendalam dan publikasinya sudah banyak terkait ini. Selain itu, Prof. Ocky juga memiliki pengalaman kinerja yang sangat bagus dalam hal pengelolaan program-program riset di kementerian. Nah, itulah undangan dan pembicara kita pada acara seminar kali ini. Dan acara selanjutnya adalah sambutan, opening speech dari Bapak Dr. Rahmadi Prasetyo. Kalau di MERO dipanggil Pak Pras, beliau adalah founder dari MERO. Jadi 10 menit selanjutnya kami persilakan untuk Pak Pras menyampaikan opening speech. In English boleh, Indonesian juga boleh. Monggo, silakan Pak Pras.
Ya, I will speak in Indonesian first. Yang terhormat Bapak Insinyur Imade Gunaje MSI, Kepala Badan Riset dan Inovasi Daerah, yang berkenan hadir pada siang ini. Sungguh suatu kebanggaan bisa ditemani oleh Bapak Made Gunaja, yang terhormat Bapak Profesor Ocky, Karna Radjasa yang menjadi kepala atau ketua organisasi riset kebumian di BRIN, yang juga adalah board di MERO. And also good morning, welcome to Ernesto. How are you? Okay.
Hi, fine, thank you.
Yeah, morning,
How are
in
you?
the morning right now in Italia.
Great to see you again.
Okay, thank you for your coming and join with us within this webinar today. Ya, ini adalah yang kesekian kali MERO mengadakan webinar, dan sebelum kita mulai, saya ingin sampaikan dulu Om Swastiastu, Assalamualaikum warahmatullahi wabarakatuh, salam sejahtera bagi kita semua. Oke, baik. Webinar kali ini sangat istimewa dan juga sangat luar biasa ya, dengan orang-orang yang hebat di bidangnya. Dan kita berharap bahwa MERO terus akan bisa berkontribusi untuk membangun jejaring, membangun iklim riset tentang kelautan, bukan hanya di Bali tapi juga di Indonesia. Ya, mungkin sebagai laporan juga kepada Bapak Badan Riset dan Inovasi Daerah, Pak Made Gunaja, bahwa MERO ya, kita 2 tahun lalu kita sempat bertemu dan Bapak juga sempat menyampaikan materi yang luar biasa waktu itu. Dan selama sekian tahun dalam pandemi kemarin, MERO juga terus dengan keterbatasan yang ada ya, kita terus mengadakan kegiatan-kegiatan baik online dan juga beberapa research yang terus juga kita lakukan. Di antaranya adalah silakan next. Ya, oke, sebelumnya sebentar, back dulu yang pertama. Ya, oke. Ya, ini adalah sebelum saya laporkan tentang beberapa kegiatan yang ada di MERO, Bapak Gunaja dan juga Bapak dan Ibu rekan-rekan sekalian peserta di sini. Walaupun dalam suasana pandemi seperti yang tadi saya sampaikan, tetapi ada beberapa pengunjung yang dalam situasi pandemi itu dengan protokol kesehatan yang sangat ketat sempat hadir. Ya, sempat datang berkunjung ke MERO. Ada beberapa perguruan tinggi yang baik ada di Bali maupun di Jawa, ada juga beberapa dari Sumatera, mereka datang, juga dari instansi pemerintah, ya dari BPSPL dan Dinas Kelautan juga hadir, lingkungan hidup dan kelautan, beberapa yayasan. Bahkan juga pada beberapa bulan yang lalu itu dari US Embassy, ya teman-teman dari US Embassy dengan 20 anak muda yang sangat fantastik, mereka datang dan selama 10 hari mereka datang dan belajar, berkontribusi untuk local development di MERO. Ya, terima kasih. Next. Ya, ini ada beberapa juga MOU yang juga sudah kita lakukan dengan beberapa perguruan tinggi, di antaranya dari Sekolah Tinggi Ilmu Farmasi Semarang, kita juga dengan FPIK Undip ya Semarang, kemudian FPIK dari Urawijaya Malang, kemudian dari FMIPA dari Sriwijaya, kemudian dari FMIPA dari Udayana, kemudian FPIK Pajajaran, serta dari Indonesia. Nah, ini teman-teman kita yang sudah ber-MOU dengan kita. Ya, mudah-mudahan barangkali di antara peserta ada yang ingin mengadakan ingin join dengan kita atau membuat MOU, silahkan nanti dihubungi dengan direkturnya Ibu Desi di sini. Oke, next. Oke, ya ini adalah beberapa riset ya Bapak Gunaja, mohon kami sekalian informasikan beberapa dalam keterbatasan suasana pandemi kemarin, kita sempat mengadakan beberapa riset terkait dengan nudibranch ya, juga dengan kolaborasi dengan Undip maupun IPB, juga kolaborasi dengan PPKI, ya, kemudian Stifar Farmasi Semarang untuk mengenai senyawa fukoidan. Dan di tahun 2022 ini kita juga terus mengerjakan untuk Torre Grant dan Joint Research dengan Center on Tropical Disease General Joint Research. Dan kalau Nudi BRIN Survey kita rutin melaksanakan terus-menerus karena kebetulan lokasi dari MERO adalah salah satu lokasi terbaik Nudi BRIN, ya, siput laut atau Nudi BRIN yang ada di Bali. Itu yang terus kita lakukan setiap minggunya. Ya, next. Juga selain research, kita juga mengadakan local development seperti Sunday Class yang juga melibatkan beberapa anak-anak kecil. Ya, di sana sangat meriah setiap minggunya. Mereka sangat menanti-nantikan kebersamaan bersama kita dengan Sunday Class, dengan pelajaran-pelajaran bahasa Inggris, praktis seperti itu, dan beberapa kegiatan lain khususnya dengan clean up, ya, ada river and beach clean up di sana. Ya, kita coba untuk mengadakan edukasi lingkungan dengan anak-anak di sana, dan sambutannya luar biasa. Ternyata anak-anak sangat menikmati membangun lingkungan yang sehat seperti itu. Juga saat mungkin Februari yang lalu ada US Embassy yang tadi saya sampaikan, ada sekitar 20 orang yang hadir, dan mereka juga tidak hanya belajar tentang kelautan atau marine, research marine, tapi juga belajar menyelam juga di sana. Selain itu juga di local development ini kita mengadakan juga plastic exchange, ya. Kita edukasi tapi juga sedikit memberikan kontribusi dengan menukarkan plastik seperti itu. Next. Ya, ini adalah Bapak Ibu dan rekan-rekan serta teman-teman yang lain apabila ingin mendapatkan progres dari kegiatan kita. Ini adalah kontak details dari MERO, baik di media sosial di Facebook maupun Instagram. Ya, silahkan diakses di sana. Ya, mungkin demikian saja sambutan dari saya. Saya berharap bahwa pada siang ini, ya, waktu terbaik di siang ini kita bisa belajar banyak dari orang-orang terbaik yang telah berkontribusi luar biasa, ya, sepanjang hidupnya dengan riset-riset terbaik tentang marine invertebrate-associated dan microorganism. Demikian, dan saya buka webinar ini secara resmi. Mudah-mudahan kita semua diberkahi dan mendapatkan pengetahuan yang banyak. Terima kasih, selamat siang. Wassalamualaikum warahmatullahi wabarakatuh. Om Santi, 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 Om.
Santi, Santi. Oke, terima kasih Pak Pras. Itulah tadi yang disampaikan progres terkini dari MERO Foundation. Oke, terima kasih. Nah, selanjutnya kita menginjak ke acara sambutan. Kami mohon kesediaan Pak Made untuk berkenan memberikan sambutan dan juga arahan kepada kami, kepada kita semua hadirin yang bergabung di room ini, kepada teman-teman peneliti, dosen, kemudian adik-adik mahasiswa. Ini generasi-generasi muda, generasi penerus bangsa yang sangat passion terhadap research. Jadi mohon arahannya bagaimana biar kita semua bisa melakukan riset yang mendukung, ya, bermanfaat untuk masyarakat, untuk bangsa dan negara. Silakan Pak Made.
Baik, terima kasih Pak Wayan. Apa yang disampaikan juga terima kasih dari founder MERO Pak Dr. Rahmadi Prasetyo. Yang saya hormati dari founder dari MERO, Bapak Dr. Rahmadi Prasetyo. Terima kasih banyak Pak Pras, sudah selalu melibatkan kami di Badan Riset dan Inovasi Daerah. Yang saya hormati Ketua MERO Foundation, Ibu Dr. Resi Kristiana, yang hadir juga pada kesempatan ini yang memberikan undangan kepada kami, dan juga Kepala Organisasi Riset, Kebumian Deputi Riset, Kebumian BRIN, Pak Prof. Ocky Karna Radjasa, yang saya hormati Pak Dr. Ernesto Mollo sebagai di Institut Biomolecular Kimia Italia, para narasumber serta hadirin yang saya hormati. Om Swastiastu.
Om Swastiastu.
Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera untuk kita semua. Namo Buddhaya, salam kebajikan, salam rahayu. Pertama-tama marilah kita tidak intinya mengaturkan sesanti angayu bahagia kehadapan Ida Sang Yang Widewasa Tuhan Yang Maha Esa, ya, karena atas asung karta warunugrahanya lah kita dapat berkumpul di tempat ini untuk melaksanakan kegiatan MERO Foundation tahun 2022 webinar dengan seri 1 dengan topik Marine Invertebrate-associated Microorganisms yang dalam suasana yang damai dan penuh kebahagiaan. Ibu Bapak yang hadirin yang berbahagia, pada kesempatan ini saya menyampaikan apresiasi dan terima kasih yang sebesar-besarnya kepada Ketua MERO, pendiri, founder dari MERO, dari Bapak Pras dan Ibu Resi, yang telah mengundang kami, Badan Riset dan Inovasi Daerah Provinsi Bali, BRIDA, akronimnya adalah BRIDA, pada hari ini dalam rapat Zoom meeting. Indonesia, perlu kami sampaikan bahwa Indonesia merupakan negara yang memiliki potensi besar sebagai marine mega biodiversity dengan jumlah keanekaragaman makhluk hidup yang beragam di laut dengan luas area perairan adalah 70% dari seluruh luas Indonesia. Tentu ini membutuhkan dukungan ilmu pengetahuan yang terus berkembang sehingga memerlukan sumber daya manusia yang dapat mewadahi dan mengelola potensi yang luar biasa ini. Sumber daya manusia yang didukung dengan keberadaan sarana prasarana dan pembimbing yang ahli di bidangnya akan menghasilkan produk kualitas serta dampak yang besar bagi generasi muda. MERO Foundation, Indonesian Marine Education and Research Organization yang didirikan di Bali pada tahun 2020 pada saat launchingnya, saya juga berkesempatan hadir acara ini dan menyampaikan beberapa materi. Nah, sekarang kondisinya sudah berlanjut lagi untuk webinar yang sesi seri 1 ini untuk memberikan dukungan berkembangnya ilmu pengetahuan bagi generasi demi generasi serta mewujudkan generasi yang berpendidikan, berpengetahuan serta sadar akan lingkungan dan potensi yang dapat dikembangkan khususnya tentang kelautan dan perikanan dengan menyediakan sarana prasarana berupa laboratorium penelitian dan para peneliti yang ahli di bidangnya. Seiring berkembangnya program pendidikan seperti adanya kampus merdeka yang mewajibkan mahasiswa belajar di instansi, MERO hadir menjadi salah satu instansi yang dapat mewadahinya dan merupakan partner kami di Badan Riset dan Inovasi Daerah Provinsi Bali. Ibu Bapak hadirin yang saya hormati, Badan Riset dan Inovasi Daerah BRIDA merupakan entitas baru pada pemerintah Provinsi Bali yang dibentuk juga hampir bersamaan ini dengan MERO. Kami diisi pada tanggal 2 Januari tahun 2020. Nah, pembentukan dan programnya akan kami koordinasikan dengan Badan Riset dan Inovasi Nasional BRIN. Kehadiran BRIDA diharapkan menjadi pengungkit pertumbuhan perekonomian Bali, organisator dan kolaborator untuk memecahkan permasalahan-permasalahan yang berbasis riset. Nah, BRIDA di Provinsi Bali ini diharapkan sebagai manajer riset. Bagaimana kita mengkolaborasikan hasil-hasil riset yang dilahirkan oleh perguruan tinggi maupun organisasi-organisasi riset atau lembaga-lembaga riset yang ada di Indonesia maupun khususnya yang ada di Bali pada kesempatan ini seperti MERO yang berbasis di Tulamben Karangasem. Saat ini beberapa daerah yang sudah atau sedang melakukan proses membentuk BRIDA, antara lain itu yang sudah ada adalah di Nusa Tenggara Barat, yang kami yang pertama di Bali, dan juga Jawa Tengah, dan juga Papua Barat. Ini yang sudah membentuk lembaga-lembaga riset dalam bentuk BRIDA. Nah, hingga akhir tahun 2022, diharapkan ada 50 brida yang ditargetkan terbentuk di seluruh Indonesia. Nah, inilah potensi besar yang dimiliki oleh Republik ini. Tentu ini merupakan peluang dari MERO untuk bisa bekerja sama dengan brida-brida yang ada di luar Provinsi Bali. Yang sesuai hal ini sesuai dengan amanat dari Undang-Undang Nomor 11 tahun 2019 tentang Sistem Nasional Ilmu Pengetahuan dan Teknologi dan Perpres 78 tahun 2021 tentang Badan Riset dan Inovasi Nasional atau BRIN. Maka BRIDA merupakan mitra penting dalam pengambilan kebijakan berbasis riset di Bali. Nah, BRIDA mempunyai tugas strategis bagaimana memberikan berbagai data dan analisis yang diperlukan dalam menghasilkan sebuah kebijakan agar seluruh kebijakan ini diharapkan berbasis riset yang komprehensif, science basis policy. Ini diharapkan bagaimana kebijakan-kebijakan itu berbasis riset. Bu, Bapak yang saya hormati, sejalan dengan hal tersebut tentu pemerintah Provinsi Bali pada saat ini melakukan transformasi struktur dan fundamental ekonomi dengan menyusun konsep transformasi ekonomi Bali. Program itu diberi nama Peta Jalan Ekonomi Kerti Bali menuju Bali era baru, hijau, tangguh, dan sejahtera. Konsep ini mengharapkan Bali dapat berdaulat di bidang ekonomi dengan mengedepankan 6 sektor unggulan. Pertama adalah pertanian dan pertanian organik, pertanian dalam arti luas. Yang kedua adalah kelautan dan perikanan. Yang ketiga adalah dari industri yang berbasis branding Bali. Yang keempat adalah IKM, UKM, dan koperasi. Yang kelima adalah ekonomi kreatif dan digital. Yang keenam adalah pariwisata. Nah, sejalan dengan program dan sektor yang dikembangkan di atas, tentu ini sudah sejalan dengan apa yang dikembangkan oleh MERO, yaitu mengembangkan riset di sektor kelautan. Tentu ini di bagian ini adalah dari sektor kelautan, perikanan diharapkan ada hasil-hasil riset dari MERO nantinya, sehingga kelautan di Bali beserta keanekaragaman di dalamnya dapat ditata dan tergali lebih bagus, apa yang menjadi potensi-potensinya dapat digali, dikembangkan lebih bagus dengan berbasis riset, sehingga dapat membantu kami untuk melahirkan suatu kebijakan-kebijakan dalam pengelolaan sumber daya kelautan secara lebih efektif, efisien, yang ujungnya adalah bagaimana kita dapat meningkatkan kesejahteraan dari masyarakat. Hal ini tentu sejalan dengan peta jalan ekonomi Kerti Bali yang diharapkan Bali ini bagaimana menata ulang perekonomian untuk menyeimbangkan struktur dan fundamental perekonomian Bali yang kembali pada keorisinalannya, keaslian, keunggulan dan ketangguhan sumber daya lokal Bali. Ini yang penting menjadi catatan bagaimana nanti MERO ini melakukan penelitian-penelitian yang memanfaatkan hasil-hasil laut untuk bisa mensejahterakan masyarakat. Inilah yang menjadi konsen kami di dalam bekerja sama dengan MERO, bagaimana hasil-hasil penelitian khususnya pada topik webinar hari ini, webinar seri 1 ini bagaimana potensi dan pemanfaatan dari hewan-hewan yang invertebrata yang ada di khususnya di Bali dapat meningkatkan kesejahteraan masyarakat. Hadirin para undangan yang kami hormati, saya kira itu yang dapat kami sampaikan secara singkat. Semoga nanti hasil-hasil webinar ini dapat jadikan kami acuan untuk melanjutkan kerjasama ini antara MERO dengan pemerintah Provinsi Bali. Dan pada kesempatan ini saya mengucapkan selamat kepada peserta dan undangan yang hadir dalam webinar MERO dari seri 1 ini dengan topik Marine Invertebrate-associated Microorganisms. Semoga apa yang dihasilkan nanti ini dalam tataran program kegiatan maupun kebijakan dapat digunakan untuk kepentingan Bali dan khususnya bagaimana kita meningkatkan kesejahteraan masyarakat Bali. Saya kira demikian beberapa hal yang dapat saya sampaikan pada kesempatan ini. Mudah-mudahan kerjasama ini bisa berjalan terus, sehingga dapat membantu pemerintah Provinsi Bali, masyarakat Bali dalam upaya mengoptimalkan pemanfaatan dari hasil-hasil laut, khususnya hewan tidak bertulang belakang, invertebrata ini, di dalam meningkatkan perekonomian masyarakat Bali khususnya, dan masyarakat Indonesia pada umumnya, sehingga masyarakat Bali menjadi lebih sejahtera dengan memanfaatkan hasil-hasil potensi laut, dan juga tetap menjaga kelestarian lingkungan yang ada dan menjaga aspek-aspek sosial masyarakat tetap terjaga. Ini suatu kearifan masyarakat Bali, bagaimana kearifan sosial masyarakat Bali, kelestarian lingkungan masyarakat Bali, dan juga pemanfaatan hasil-hasil potensi laut untuk masyarakat Bali, sehingga dapat meningkatkan kesejahteraan masyarakat Bali. Saya kira itu beberapa hal yang kami sampaikan pada kesempatan ini. Sekali lagi terima kasih banyak kepada pendiri MERO dan juga Ketua MERO Foundation yang sudah konsisten mengajak kami untuk melakukan kerjasama ini. Sekian dan terima kasih. Saya tutup dengan permasanti. Om Santi 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 Om. Wassalamualaikum warahmatullahi wabarakatuh. Selamat siang, salam sejahtera untuk kita semua.
Nggih. Nggih, suksema Pak Made, terima kasih. Thank you so much. Jadi secara singkat saya bisa tangkap bahwa nafas dan semangat Brida itu sesuai dengan kami MERO, sehingga besar harapan kami juga di MERO untuk berharap bisa tetap bekerjasama dengan Brida dalam mengembangkan riset di Bali dan juga untuk memajukan riset di Indonesia. Oke, terima kasih Pak Made sekali lagi. Acara selanjutnya adalah itu overview dari MERO Foundation dari chairman-nya. Namun sebelum itu seandainya Pak Made memiliki kegiatan lain yang sangat penting, agenda penting berikutnya, kami persilahkan. Namun jika Pak Made berkenan untuk ikut bergabung dengan kami sampai acara ini selesai, dengan senang hati kami bisa bersama Pak Made di sini.
Terima kasih Pak Wayan, terima kasih banyak. Saya mohon izin nanti ada staf kami yang akan mengikuti kegiatan ini karena ada saya penugasan lain. Selamat berwebinar, mudah-mudahan ini berjalan lancar ya. Terima kasih Pak Wayan dan Pak Pras dan Ibu Resi.
Terima kasih Pak Made Gunaja.
Terima kasih Pak Made. Terima kasih
Pak.
Okay, all right, ladies and gentlemen. So next, we are going to have a speech from the chairman of MERO Foundation, and then we start now using full English. So please, the chairman of MERO Foundation, Resi, you have 10 minutes to deliver your speech. To Ernesto and friends, we are now back online. So just be prepare your slide soon after the speech from the chairman. Okay, Resi, you have 10 minutes for your talk. Please go ahead.
Yes, thank you very much, Pak Wayan. Maybe I will also use bilingual today because we have a mixed participant. So first of all, I would like to say thank you very much and welcome to our speaker, Pak Dr. Ernesto Mollo from Italia, and also MERO Foundation team, Pak Pras, our founder, and also Pak Wayan, our moderator today. We are as board member, and also Prof. Ocky later will join. And also I would like to thank you very much for the speech from Bapak Insinyur I. Made Gunaja. Terima kasih Bapak matur suksema atas kehadirannya yang selalu senantiasa bersemangat untuk mendampingi kami MERO Foundation mulai dari tahun 2020. Dan kemudian saya juga mengucapkan selamat datang untuk seluruh undangan pada siang hari ini dan juga seluruh peserta, para dosen maupun peneliti dari BRIN dan dari instansi lainnya dan juga mahasiswa yang sangat kami cintai. Terima kasih sudah hadir dan menyempatkan waktunya 2 jam ke depan untuk belajar bersama dan berdiskusi di acara MERO Foundation ini. Untuk selanjutnya saya menghimbau apabila tidak ada kepentingan yang lain untuk tetap mengikuti webinar ini sampai di akhir, karena kita akan didampingi oleh para pembicara yang hebat, sehingga sebagian dari waktu nanti akan ada banyak diskusi. Jadi silakan banyak memakai waktu untuk saling bertukar pikiran maupun ide. Jadi kita akan banyak berdiskusi. Untuk selanjutnya saya akan melanjutkan overview sebentar, mungkin 5 menit ke depan. Saya izin share screen ya. Mungkin saya bisa share screen sendiri, panitia. Ya. Oke. Ya, sebelumnya saya ucapkan juga selamat siang, Assalamualaikum warahmatullahi wabarakatuh, Om Swastiastu, salam sejahtera bagi kita semua. Ya, di sini, today I would like to introduce also to Ernesto. Maybe this is your first time to come and join in MERO Foundation. So we just established in 2020, we are focused in education and in research, especially on marine base. And then MERO Foundation also provides the facility, laboratory, dormitory, and also restaurant, and also diving for the research in marine education, for the local development, for all the student, lecturer, or researcher in Indonesia or in international. So we try to develop our research, our strength research, and also collaboration, and also how to develop our potential in Tulamben, Bali. This is the overview of the habitat and the location of MERO Foundation. So we are in front of the sea. In here, we have so many kind of, especially marine invertebrate, such as tunicate, coral, and also sponge and nudibranchs. So today we would like to hear also your speech about the ecology of the marine interaction. And in here we also have the local people, local village, and also children who always interaction with us and learning with us every week. So this is our short term and long term. For short term, we would like to educate the local people and also national, and we would like to provide the research for student, lecturer, and also researcher. And the long term, we would like to be a central of research and the education based on marine. This is the key action from 2020 to 2022. This is what we have done and we are going to. So we start in 2020 to build the laboratory and facility, and also we recruit some human research, and also we try to promote collaboration, and now we already become a place for the internship and research for the researcher and also the student. And now we try to develop the strength of the research, become more impactful, and also more substantial of the research focus. So this is our laboratory facility. We have camera area, microbiology room, including bacteria room, and also fungi room, and also DNA extraction, PCR, chemical room, and also SEM, scanning electron microscope, and also the next generation sequencing. This is the overview of our research. We try to observe some invertebrate, microbiology, and also another activity. This is some of the sample of our product. So we try to identify the radula of nudibranch by SEM, and also some part particle, and also sponge. On the other hand, we also have the own database in MERO Foundation about the collection of marine bacteria or actinobacteria. So we collect all of them and we save all of them. In the future, we can develop about the potential of this marine bacteria. We also collect the database about the diversity of the marine organism, because there are so many marine organisms that's not described yet. So some of them are from hydroid, and then sponge, nudibranchs, tunicate, and anything else. And then this is our future prospect. First, we would like to develop in the pharmaceutical. So we start to collaboration with some pharmaceutical university and looking for the industrial. This is the sample from dolastatin. Dolastatin is the compound from nudibranchs as anti-cancer. So we would like to develop more. And also in the molecular parts and the conservation in the sea. This is our picture, our photo when we took during diving. We see some of animal also trapped in the net. And in the local development, we would like to try also about the plastic. So how they care about the plastic and how they can use the plastic to become something. So this is I always explain about this, our service and about our research in about antibacterial, microplastic, DNA barcoding, and also molecular, and also the extraction of the bioactive compound from the sea, and also how to characterize the morphological appearance from the marine invertebrate, and also another topic area. And this is the picture. This is so cute. This is the kids from the village. Yeah. Not only research, we would like also to transfer this potential to the very young generation. So from the senior high school, this is we provide this program by request. If they would like to use the SAM or they would like to study about biology, so they can use this instrument. So this is what we have done. We support the school around the village, and then the activity by request, and also the local development. This is all of the activity that we already done. It's about internship, and then research, collaboration, and also volunteer. So we would like to welcome all of you. Yeah, so that's all about the explanation from me. So thank you very much. Saya juga mengucapkan selamat datang untuk seluruh peserta yang baru saja bergabung. Jadi masih belum terlambat, silahkan bisa bergabung sampai di akhir nanti ya, karena kita akan ditemani oleh 2 pembicara yang ahli di bidangnya. Okay, so that's all from me, Pak Wayan. So I give the time back to you. Thank you very much for your attention.
Okay, great. Thank you, Resi. Can you hear my voice?
Yes, Pak Wayan,
okay,
very clear.
Good. Thanks. Okay, that's all detail about MERO, where MERO is and what we have today. So the picture that's shown at the beginning of the slides by Resi, it might not be suitable now because now we have another lab, another building already in MERO established brand new buildings, but still empty. We are waiting for scientists and also instruments to come very soon. Okay, all right. So next is the presentation from the first speakers. Ernesto, are you ready to talk? Okay, thank you.
Yes.
Nice to see your face again. So Ernesto was here back in 2018, I think. He spent a few days somewhere in next to MERO Foundation. At that time, the building was not ready yet. MERO just only an idea at that time, but now is already here.
Great.
So MERO is already born and all the facilities are available now. So for your information, Ernesto is one of the, if I can say, one of the best marine ecologists. He has been doing very detailed research about marine organisms and the relationship between the chemistry and ecology of especially the nudibranchs, the sponge, and so on. And his recent paper is just available online. It's about smells and tastes of marine organism. This is a very recent theory and philosophy about taste and smells among the marine organism. So this is kind of unifying chemosensory theory. Okay, he will talk about this later on in more detail. So if you have any question later on, just keep writing and keep it for the moment. Then we will discuss it during the question and answer session. Okay, Ernesto, you have 40 minutes to talk, and you might need to introduce yourself first to the audience. So we have now 62 young students, scientists from all around Indonesia joining with us. Most of them are coming from university. They are lecturers and also a scientist. And the rest are coming from the government laboratory or something like that. So, so yeah, various background, but all of them are interested to hear recent progress in your research area. Okay, so if you would like to share your screen, please do it now.
Yes,
and then
let me try.
Yes, and then you can talk now.
So
yes, I can see your slide. And if it's possible to make it in slideshow mode, then it would be great.
Let me try.
Yes.
Okay, you can see.
It's not yet in. Okay, all right. So Ernesto, once again, I would like to say that this is fantastic picture. I know that you created this picture. So Ernesto is a talented art actually. So teman-teman, kawan-kawan, beliau ini seniman juga nih ternyata. Karena beliau sering buat grafik-grafik yang seperti ini. Okay, all right, Ernesto. Please introduce yourself and start your presentation. Ah, what's that? Oh, the token. Lovely. Yes.
Yes.
Okay. All right.
So good morning. Thank you, Wayan, for your kind introduction. Can you hear me?
Yes, it's clear.
Yes,
please.
Thank you. So good afternoon to everyone. It is really a great pleasure for me to participate in this series of seminars. And I am very grateful to the MERO Foundation for inviting me. My purpose today is to highlight the crucial ecological rules of small organic molecules. They are commonly called secondary metabolites. And in particular, I would like to show you how we can use chemistry as a magnifying glass to explore marine ecology. But first, let me give you a brief introduction to the secondary metabolites. Here you can see some examples. They are natural products not directly involved in central metabolism. However, they are very important because they are known to mediate fundamental biological processes such as reproduction, competition, defense, and so on. They are often species specific, and many of them were originally investigated because of their medicinal, perfumery, culinary, recreational, and other special value to humans. However, just recently, in last decades, increasing attention has been paid to their function in nature. However, we are still far from full understanding the relationship existing between their ecological and pharmacological properties. So this is the result of a sort of compartmentalization of disciplines, which are they are different bodies of knowledge, chemical ecology and drug discovery. They are separately developed, each having its own publication outlets, while more integration of the results from both areas could allow one to address issues that reach beyond the disciplinary skills of individual scientists. So the challenge is to unify these two disciplines. However, during my career, I especially focused my attention on the secondary metabolites that are continuously isolated from marine organisms, marine natural sources. The biological function of this compound, which in most cases remain to be clarified, was my main interest. I personally performed many sampling campaigns around the world to find the novel metabolites from the sea. Here, I am in company with the Chinese colleagues and friends along the coast of the South China Sea. Here we are in Mexico, here in Egypt, in Antarctica, and here in Venezuela, and here in India. In this picture, I was quite young and still with long hair, but I was enthusiastic at that time, especially in studying these wonderful animals, the heterobank mollusks, and in exploring their defensive strategies and their chemical language. Actually, secondary metabolites play critical roles in the life of heterobanks. They use chemicals as defensive allomons to protect themselves from predators. They follow chiromones to identify their favorite prey, but also they use pheromones to communicate with the conspecific. It is a kind of language widely employed in nature, and not only by mollusks. By looking at the letter, you can see in the literature that also in plants, bacteria, and in almost all living organisms, chemicals play a role as words of a complex language. During my studies, I have been especially interested in decoding the words of this language. Although chemistry is not strictly a language, the metaphor is useful to describe a situation in which sensory signals symbolize and communicate a meaning, influencing the behavior of the receivers. However, it has been pointed out that chemosensory, acoustic, tactile properties, along with visual appearance, can be all taught as elements of an aposematic gestalt that a predator make use to key out a potential prey item in its whole configuration, perceptual configuration. Another example would be crypsis, that can involve simultaneously visual, chemosensory, and even auditory concealment. Aposematism is a well-known case of communication occurring via multiple sensory channels. This phenomenon has attracted the attention of prominent biologists and was originally intended by Poulton as a visual appearance denoting something unpleasant or dangerous. A typical case of aposematism is that of the yellow frog Phyllobates terribilis. A single individual of these brightly colored animals contain enough batrachotoskin, this molecule, to kill 2,000 mice. In this case, the yellow color warns the predators, allowing them to immediately identify the dangerous prey. In nudibranchs, however, the association between chemical and visual message is particularly frequent. For example, when I collaborated in a study carried out by Australian and Indonesian colleagues, I had the opportunity to observe that a group of similarly colored nudibranch species belonging to the genus Chromodoris sequester and store the unpalatable and toxic compound latrunculin from a sponge for defensive purpose, implying that this group of nudibranchs not only share a common color pattern, which is widespread in the whole Indo-Pacific, but also a common chemical signal. They form a putative Mullerian mimetic cycle. Actually, the yellow, white, orange, and black color patterns of these nudibranchs is very similar to the color patterns of the heliconid butterflies studied by Fritz Müller in South America. These similarly colored species mimic each other. These are honest warning signals because they are unpalatable. And they share this signal to their mutual benefit, given that predators learn to avoid all of them with fewer experiences with members of any one of the relevant species. In this case, instead, this is the case of the Batesian mimicry. In this case, a palatable species might gain protection from the predators by resembling unpalatable species, which is the honest signal. So they are harmless. However, predators avoid them. It is like the term people use in the poker, playing the poker is a bluff. They make a bluff. So, however, the existence of a Mullerian mimetic circle within nudibranchs, which are often called the marine butterflies, was first proposed by Professor Giandomenico Rossi in the 70s for the group of blue, white, and yellow Mediterranean and Atlantic nudibranch species. At that time, he was in contact with my professor, Guido Cimino, who was a chemist, and they started collaborating, searching for the chemicals in these animals. And I was a student at that time, and I started working on this topic. And we confirmed that all studied species were provided with defensive chemical weapons. And even in the case of Felimare fontandraui, in this case, we found this nudibranch lack some the mantle dermal formation. These are some defensive structures that are actually the reservoirs of these tasteful metabolites. They are placed on the more exposed part of the body of the nudibranchs. In this case, the Felimare fontandraui lack these glands. So some colleagues proposed that it could be a Batesian mimic, so that it is not toxic, not the feeding deterrent. However, in these animals, we found the compound Tavac pallescensine, and we found that it's accumulated in the border of the mantle of the nudibranch at concentration much higher than those that were able to deter predators from feeding in our assays. Therefore, with its defensive chemical weapons, also this nudibranch is an effective member of the Mullerian cycle we anticipated, that was anticipated by Ross. We are still searching for some Batesian mimic among nudibranchs, but till now they are all chemically defended. So the assessment of the feeding deterrents of the compounds found in the nudibranchs were carried out thanks to the use of this food deterrence test on crustaceans that we developed on 2008. But that Wayan also adapted to other shrimps commonly found in Australia. These kind of shrimps are widespread all over the world, different species, of course, but they are very suitable to this experiment. We can prepare the food by adding some coloring, food coloring, to look if the food come inside in the stomach and in the gastric meal of the shrimps. In this case, we have a positive response that the shrimps feed on. Otherwise, the spot is not present. And we can repeat this experiment in a series of individual replicas to have the statistics. In this way, the same assay was used by us to investigate the importance of the anatomical distribution of the so-called mantle formations that are located in the more exposed part of the body of most nudibranchs. Given that these structures usually lack a duct system connecting to the outside, the mechanisms for the exudation of their content was unclear, and as well their adaptive significance. Our hypothesis was that they are adapted so as to be preferentially attacked by predators. We detected extremely high local concentration of secondary metabolites in the MDFs of 6 species belonging to 5 Chromodoris genera collected along the Chinese coasts. The compounds were purified and by chromatographic methods, and subsequently they were evaluated for their feeding deterrent properties on shrimp, obtaining dose response curves. We found that only distasteful compounds are accumulated in the reservoirs at concentration that far exceed the values corresponding to the maximum deterrent activity in the feeding assays. Finally, to evaluate shrimp's ability to produce damage to the anatomical parts where the MDFs are located, we offered mock, false, unprotected nudibranchs made of squid. I made these models of nudibranchs. This is frozen squid, and I sculptured and made these little sculptures. They resemble the condition of an uncolored mantle edge of a dorsal, also of a dorsal finger-shaped appendage and the border of mantle, and we offered these mock nudibranchs to the shrimps. As expected, damage to the models was produced selectively in the position where the MDFs in the corresponding nudibranchs are located. All these findings supports that as a result of a long evolutionary process that has progressively led to the accumulation of defensive chemical weapons in localized anatomical structures, the extant chromodorid nudibranch remain in place when they are molested, retracting respiratory and chemosensory organs, but offering readily accessible parts of their body to predators. When these parts are masticated or wounded by predators, and here you can see a ceratosoma lacking the dorsal horn because it was bitten by a fish, breakage of the MDFs in the mouth of the predator result in the release of these tasteful compounds at extremely high concentration in a way that maximizes their repugnant impact. We used a metaphor to describe this, the metaphor of the Trojan horse. In a sense, the nudibranch offers packages containing high repugnant chemicals along with parts of their bodies to the predators, as a defensive variant of this strategic theme of the Trojan horse. You know the myth. If you okay, back to the linguistic metaphor that chemicals are words of a complex language. Some chemical messages can have different meaning according to different receivers. In this case, they act as words of different languages that sound similar, but they differ significantly in meaning. They are commonly called false friends of a translator. Let me show an example from my experience as a translator, chemical translator, but in chemical ecology. These Furanus sesquiterpenes are produced by several terrestrial plants, including the white turmeric. We also found them in the Mediterranean octocoral Masella dvarzi and in its specialist predators, the nudibranch Tritonia striata. Here you can see the nudibranch and its egg masses laid on the coral. On the right, there are also the developing larvae of Tritonia striata. We first observed that all these three compounds were toxic when assayed for their general toxicity on brine shrimps in a dose-dependent manner. Then we evaluated the feeding deterrence properties of all three compounds against the generalist shrimp Palemon elegans. Here you can see on the left the shrimp accepting the control food. In this case, a red spot evidently appears in the stomach of the shrimp. On the right, you can see instead the rejection response to a feeding deterrent dose of the compound. Also in this case, we found that all three compounds produce rejection responses in a dose-dependent manner. We did not observe, however, any lethal effect of the compound when ingested by shrimp. Just in some cases, we observed emetic events in the shrimps that accepted moderate doses of the compound. And here you can see what I mean. The vomiting shrimp. This observation led us to perform an avoidance learning experiment to see if the shrimp, after this negative experience, remember the bad experience. And we made and here you can see the full experiment. So in short, this experiment led us to demonstrate that all shrimp that had previously experienced emetic events, vomit, significantly rejected the treated food during a subsequent administration, having just touched it, while this then significantly accepted non-treated food. The studied compounds thus turned out to act simultaneously as toxins, aposematic cues, and avoidance learning inducers. Overall, the main message they convey to potential predators is, please do not eat me, I am dangerous. All three compounds instead convey a completely different message to Tritonia striata. This is why they are also the false friends. They are present in the mucus covering the octocoral. In spite of this, we observed the nudibranch engulfing the polyps of the cnidarian. This is a very nice picture from my colleague Guido Villani. And you can see the polyp in the mouth, in the buccal bulb of Tritonia striata. This evidence strongly supports that the nudibranch obtain the metabolites from its prey and accumulate them in its mantle, and that the compounds, in the case of the nudibranch, they do not represent a warning message at all. Instead, they possibly act as chiromones, indicating the presence of the suitable food. In general, a chiromone is a semiochemical emitted by an organism which mediates interspecific interaction in a way that benefits an individual of another species. Recognizing the proper food is crucial for the survival of monophagous species such Tritonia striata. The compounds are also present in the larvae of the cnidarian, the planulae, and here we also observed the nudibranch engulfing. You can see it start the video. Look, oh, sorry. So you can see on the left in the yellow square, look, the nudibranch engulfing the larva. Hey. And these larvae are full of these metabolites. So overall, our results led us to propose. Let us to propose multiple ecological rules of the isolated odorant Furanus sesquiterpenes that convey different messages depending on the different receiver organisms. thus acting as words in two languages that look or sound similar, but differing significantly in meaning. They can act as both defensive warning message and toxic chemical weapons if directed to potential predators or prey. But they evidently do not deter Tritonia striata, the specialist predators, from feeding. On the contrary, in the latter case, the three compounds possibly act as pheromones and indicate a food source for the receiver, which is able to handle and reuse them in self-defense. However, the toxicity of the compounds to brine shrimps is also suggestive of their involvement in mechanisms of predation as part of the alimentary strategies of Macella advanci in nature, helping to capture dietary zooplanktonic crustaceans, for example, copepods, that come into contact with the mucus layer covering the octocoral, and which lose their ability to escape owing to intoxication. Given all these premises on some of the chemosensory words spoken in the marine environment, here are some example of words in Europe. We are now ready to approach a critical theme in a study on outdoor products when they actually act as loan words. But what are loan words? They are words taken from one language and used in another language. Here you can have some common examples of words that designate new exotic foods that move from one place to another in Europe, as much as invasive species colonize new environments. This last topic is particularly critical for the Mediterranean Sea, that is an hotspot of marine biological invasions. During my career, I have had the opportunity to study the chemical composition of some among the most invasive species in the Mediterranean. One of these is Aminea cyanomarginata. Was the first to find this cephalaspidean in the Mediterranean along the coast of the Gulf of Corinth in Greece in 2001. But before going on, I would recall that indigenous Mediterranean amineas, these are our amineas, Italian aminea, use alarm pheromones for defensive purpose that indicate the presence to a danger to conspecific. These compounds are non-toxic, and they are involved they are not involved in interspecific communication. They just communicate among individuals of the same species. So the compounds are non-toxic. They show to conspecific that there is present a danger, and the coloration of these animals are cryptic. In Aminea cyanomarginata, instead, we identified a brominated tetrahydropyran we previously found also in Aminea cymbalum I collected along the Indian coasts. Interesting, both animals are brightly colored, sensibly differing from the Mediterranean congeneric species. They speak another language, completely different language. Chemically. However, to assess the palatability of the main metabolite of Aminea cyanomarginata, we first quantified its natural volumetric concentration in the mollusk. Then we assayed the compound for its palatability to shrimp. The compound turned out to be significantly active in deterring shrimp from feeding at its natural concentration. This led us to propose that aposematism would have increased the likelihood of the establishment of these mollusks in the Mediterranean, especially during the first phase of colonization, when the sacrifice of a few colonizing individuals could also have prevented the establishment of an active population. The Mediterranean species instead rely on alarm pheromones for defense in a defensive strategies that imply the sacrifice of a consistent number of slow-moving animals. So possibly this aposematis is more effective, and thus it favored the migration of these new species in the Mediterranean. In the Gulf of Corinth, during a night dive, we also found the sea hare Siphonota geographica. The first study led us to isolate the degraded spheros from which a defensive rule was implied by the fact that they were selectively localized in the skin of the animals, exhibiting, however, strong structural analogies with bioactive molecules found in other anasphideans from different geographic areas. But when we approached the study of the visceral part, the digestive tube, we found the known flavones that are widespread in terrestrial plants, but were never found in a marine seagrass. Together with a new class of secondary metabolites, we call it siphonosides. The presence of these metabolites in the digestive system suggested us their possible dietary origin from a seagrass, from a plant. So we analyzed by a microscope the plant fragment in the stomach of the animal. And this led us to identify the seagrass Alophila stipulacea. This plant is invasive and was recorded on 1895 for the first time in the Mediterranean, a few years after the opening of the Suez Canal. The chemical study of a sample of this plant collected in Greece during a further campaign led us to isolate the same compounds we found in each mollusk separately analyzed. Our finding was indicative of a close dietary relationship between the animal and the plant, and led us to propose that the migration of the prey would have facilitated the migration of its specialist predator. 100 years later, most likely acting as a chiromons, attractant chiromons. On the other hand, flavonoids have a number of important functions in plants, acting as regulator of symbiotic interaction with the microorganisms, maintaining a redox state in cells, and participating in protective interaction with the protective strategies against herbivores and pathogens, as well as against abiotic stress, such as UV radiation and heat. As a result, these compounds could confer to Alophila stipulacea a competitive advantage over native species, sensibly contributing to its success as invader in the Mediterranean Sea. It would therefore be appropriate to carry out further studies to confirm this hypothesis. And now, I don't know if I have time, but I would like to conclude with a passage from our recent paper that Wayan mentioned just a few minutes before. This is from our recent paper we published in the Quarterly Review of Biology. And the paper is already available online and at this link. And I would like to highlight that the knowledge of the actual spatial distribution of the chemosensory signals in different environments can greatly contribute to a better understanding of the heterogeneous distribution of species in a variety of habitats, which cannot be framed within the traditional taste, smell dichotomy. This is just, however, one aspect of this new perspective on chemosensation that led us to propose the unification of all chemosensory modalities. But this is another story that I cannot deal with here for reason of time. So let me hope, however, that there will be other occasion that we will another opportunity to discuss this topic together within the MERO webinars. For now, with this figure from that paper, let me thank you for your attention.
Thank you. All right. So Thank you very much, Ernesto. So it's
Thank
just
you.
wonderful pictures and videos. We like it very much. Okay, Ernesto, would you mind if I talk in Indonesian to the audience a little bit to just
Of course.
explain what is the main main of your some point of your talk? I need
Yes,
to
and
explain to the
please,
audience. Yes.
thank you very much. And please translate if there are some questions.
Okay. Yes. Okay. At the moment, you can drink your cappuccino while I'm explaining to the audience. And,
Okay. Thank you.
Okay, because it's early morning, so I suppose you are having
Yes.
breakfast at the moment. Okay. All right.
Yeah.
Bapak Ibu
Okay.
peserta seminar. Oh ya, saya ingin menyapa Prof. Ocky dulu. Prof. Ocky, selamat sore.
I cannot see. What is this?
Prof. Ocky, selamat sore. Oke.
Buongiorno, Ernesto.
Buongiorno.
Ernesto, he's the second keynote speakers today. He's a Professor Ocky from the National Research and Innovation Agency. So
Pleased to meet you.
Yes.
Nice to meet you too.
Oke, alright. Yap, Prof. Ocky, terima kasih atas kesediaannya. Ini luar biasa di tengah-tengah kesibukannya masih bersedia meluangkan waktu untuk kita semua.
Iya,
Oke.
lagi rapim saya sempetin.
Luar biasa. Oke, Prof. Ocky dan kawan-kawan semua yang masih setia di room ini, Ernesto Mollo itu peneliti senior ya di bidang ekologi kimia. Kita sudah simak tadi, luar biasa tampilan slides dan video-videonya. Intinya begini. Saya sempat 2 kali ke labnya dia di Italia itu ya. Nah, kunjungan saya pertama itu saya berharap labnya Ernesto itu ada di sampingnya markas sepak bolanya Inter Milan ya, punyanya Pak Erick Thohir atau sampingnya ya Valentino Rossi gitu. Ternyata jauh. Jadi labnya si Ernesto ini ada di Napoli di selatan Italia dan di pinggir laut Mediterania. Jadi langsung dia lab alamnya ada di sana. Dan pertama saya di sana langsung diajak nyari udang, luar biasa itu. Pertama karena dia memang basicnya ekologi riset itu dia ujinya pakai udang dia di sana, udang Palemon ya, yang banyak ada di sana itu. Nah intinya dia itu concern ke bagaimana hubungan peran-peran senyawa kimia itu di branch itu di ekologi di lingkungannya dia. Kalau nudi branch itu barangkali nama lainnya adalah apa ya itu ya, siput telanjang ya. Kalau Kompas pernah menulis kemarin siput telanjang atau kelinci laut untuk spesies tertentu yang ada kupingnya ada kupingnya naik itu ya, disebut kelinci laut juga gitu. Atau butterfly of the seas juga sama itu di kupu-kupu laut juga sama. Nah intinya itu kan makhluk kecil yang lemah gemulai, warna-warni yang cantik sekali, sering dipakai sebagai objek fotografi bawah laut. Namun jangan dianggap sederhana ya, bahwa dia ternyata mampu mempertahankan diri dengan chemical defense ya, jadi kayak pertahanan kimia dia punya. Maka dari itu jarang banget terekam atau teramati nudi branch itu dimakan sama ikan. Nah, kenapa bisa begitu? Nah, karena dia memiliki pertahanan kimia, chemical defense. Dia secara evolusi menaruh pertahanan kimianya, senyawa-senyawa kimia itu di pinggir badannya, di rimnya. Tadi sudah ditunjukkan di fotonya. Ada kayak klepon. Kalau di Bali ada klepon, jajan yang bulat, yang dalamnya ada gula merah. Nah, itu ya. Nah, jadi kalau misalkan nudi branch ini diserang sama ikan, predator, itu yang dimakan, digigit itu pasti kena rimnya dulu, pinggirnya itu. Nah, di sanalah racunnya ada, sehingga dimakan oleh ikan, lalu pecahlah di dalam mulut ikan, akhirnya dimuntahin. Nah, setelah itu ikan akan belajar learning avoidance ya, dia akan belajar bahwa ini makhluk kecil ini jangan dimakan, nanti dia kasih tahu anak cucu dan seterusnya, sehingga secara evolusi nudi branch itu memiliki itu. Bagaimana cara menentukan senyawanya? Nah, dari hasil penelitian itu senyawa murni betul, single compounds dan konsentrasinya tinggi banget. Jadi volumetric konsentrasinya itu bisa diukur dengan menggunakan NMR. Nah, kemudian di lab bisa kita uji dengan menggunakan udang. Tetapi zat-zat kimia itu atau ekstrak-ekstrak dari spon dan sebagainya itu harus dibuat dalam bentuk spaghetti. Ya, jadi Ernesto ini menemukan cara bagaimana membuat spaghetti. Jadi zat kimia itu dicampur dengan squid, kemudian apalagi zat lain, dicampur jadi spaghetti, itu dikasih ke ikan dan seterusnya. Ke udang juga. Itu tadi ada udang muntah, video udang muntah, luar biasa. Saya pertama kali lihat itu. Betul-betul muntah udangnya karena dia tidak mau mengingest itu zat-zat kimia itu. Nah, secara umum itu riset-riset yang dia lakukan. Jadi konsep-konsep baru juga ada, hasil-hasil terbaru. So okay. So itu saja yang bisa saya highlight dari apa yang dia sampaikan tadi. Kalau teman-teman misalkan bisa langsung bertanya dengan Ernesto dalam bahasa Italia, silakan, itu akan lebih pas gitu ya. Kalaupun tidak, ya bahasa Inggris juga boleh, bahasa Indonesia nanti saya terjemahkan. Oke, next saya buka untuk sesi diskusi ya, Resi ya, kita diskusi. Ernesto ada diskusi, setelah itu Prof. Ocky diskusi lagi ya. Oke, teman-teman yang punya pertanyaan silakan. Teman-teman MERO tolong dibantu saya untuk mengingatkan, barangkali ada yang sudah lebih dulu mengajukan pertanyaan kepada Ernesto, silakan. Ernesto, now we are coming to the discussion session, so please be prepared to receive the questions and comments from the audience. Okay, so yeah, okay, everyone. If you have any questions about nudibranchs, chemical ecology, please just raise your hands and then introduce yourself and just deliver your questions. Okay, in the chat room, please help me to see if there is a question. Okay, all right. So, questions? Okay. All right. So, while waiting for the questions, I just need to make comment, Ernesto.
Thank you.
Now you are you have been diving in many places in the world, in China, in India, in South America. So before entering the retirements, okay. So I would like to I would like to invite you to come to Bali first. Okay, so to Bali, because this is like an area that under investigated. So plenty of nudibranchs available here, and then we have the lab as well. So okay, so
yes, it will be great, really.
Okay, yes, I need you to train the next generation of scientists in Indonesia, the young scientists, how to carry out research on nudibranchs, how to make spaghetti, how to make a lovely nudibranchs pictures, and how to make a video of eating of a shrimps vomiting and so on. So this skills is just absolutely fantastic. And then currently it's a bit difficult to find someone expert like you in Indonesia. And MERO now have a facilities to carry out that kind of research. We have built a new lab, and then this is for aquariums and also other studies. We also have SAM, and soon we're gonna have NGS sequencing for DNA. So it's gonna be fantastic.
This is great. This is really great. Also because still we don't know if there are some symbiotic bacteria in the gut of these nudibranchs that contribute to the synthesis of some of these metabolites. So it will be very interesting to analyze the microbiota of these nudibranchs.
Okay, fantastic. Yes, because microbial is one of the main core of our business as well at MERO. The main research topics in MERO, we are very strong in growing bacteria from marine environment, including from the internal organs of nudibranchs. Okay, lovely. So if you were in Bali doing research, then you can have a holiday as well at the same time. So in 1 year, you're doing research and holiday at the same time. Anyway, all right.
Great. Yes.
Okay.
Thank you so much.
Okay, anyone?
Let me hope.
Yeah, sure. Okay, there's a question from someone here. Ernesto, I'll read out the question for you, okay?
Yes.
Okay. As we know, nudibranch has a specific food preference, such as hydroids, sponges, algae, and bryozoan. Most often, we find nudibranchs in their food area. We also find phenomena, sometimes they appear so many in the sea, and sometimes we did not find them. What is your opinion about the phenomena? So this is from RK Risi Christina. Okay, all right.
Oh, okay. This is. Thank you for this question. It is very interesting. However, you know the koala in Australia. Koala eat eucalyptus. The koala is a specialist. Just eat eucalyptus, and for the koala is crucial to identify its food. Nudibranchs are the same. Nudibranchs often are monophagous or eat just a few sponges. So for them, it's extremely important to find the sponge. Some of the, and how they can find their prey. They do not have eyes. They just have some sensors that allow to distinguish the light from the day from the night, but they cannot recognize with eyes their prey. So they have to follow chemical tracks on the bottom. So most of these compounds are lipophilic. They are not soluble in water, so they adhere to the surface. And these animals follow the track to find their own food. This is very interesting because also to organize experiments, because if you are able to isolate the metabolites present in the prey, in the food, you can organize some experiments, making some multi-choice experiments, or using an epsilon tube, where you put the nudibranchs in an aquarium, and you have a tube, an epsilon tube, and in one aquarium on the right, you can put the sponge or the single compound, and on the left, nothing. And you can see the nudibranchs if follow this. But this experiment in the past were made by dissolving the compounds using DMSO, some vehicle, because the compounds are not soluble in water. After our publication in June on the taste smell, I think we should start making some experiments by placing the compounds on the bottom, adherent to the bottom of the aquarium, in some surface, in some way. We have to think, to imagine these experiments. Of course, these experiments, you can do these experiments of course, in Bali, in the place where you find the nudibranchs, and I think there is matter to organize many experiments. However, these compounds are chiromons, are molecules that give another advantage to the nudibranchs to find the prey. However, there is another point very interesting. You know that many of these nudibranchs, the eggs, from the eggs of these nudibranchs develop larvae that are pelagic. They are planktonic. They move in the water. At one time they make the settling, they metamorphosize and pass to the benthic life and start moving on the surface. I suppose, and I'm not the only one, that there are signals, chemical signals in the prey that can induce the settling, the metamorphosis. They indicate that the place is suitable to metamorphosize, to settling. Then, sorry for my English, but I hope you can understand. And also this kind of experiments, I think we should try to organize, to see if there are compounds from the prey that stimulate the metamorphosis, that induce the metamorphosis in the larvae of the opisthobranchs. So there are very few evidence on crude extracts, but we can work on the purified compounds.
Okay.
I don't know.
Okay,
If I,
all right.
Every question, from every answer, you can find other questions.
Yeah, sure. Okay. All right. Thank you, Ernesto. That's the response from Ernesto. Okay, Resi. So next from
Yes,
Dwi
thank you
Imam.
very much, Ernesto.
Okay, from Dwi Imam. There's another second question for you, Ernesto. I'll read it out for you. So the bottleneck for supply is always a problem at marine natural products. So what approach for that? Could be extraction the compounds without harm or kill the subject. So I guess he asking about when we're dealing with drugs discovery development, something like that, when we need a lot of extract to start the research. So how to solve this problem?
Yes.
Do you have any
so this
comments?
is,
Yes.
Yeah, this is always a problem, of course. Although first, chemists working on natural products must work on small amounts of biological materials to make less impact on the environment. However, of course, if you find some compound particularly interesting, of course, a main way is to organize a project to make to produce the compounds artificially, to make the synthesis, the total synthesis of this compound. This is the way. And however, I have to say that in some case, for example, in the Mediterranean, when if we consider the invasive species, for example, they produce huge biomasses. This biomass you can use, because if you remove this undesired biomass from the Mediterranean, this is good for the environment. So in this case, we do not have this problem for the invasive species. But in most cases, when we talk about nudibranchs, and of course, we cannot use nudibranchs to obtain huge quantity to be employed in pharmacology or to synthesis. This is the way.
Okay. All right. Thank you for the quick response on that. So, okay. So I think there's no more question in the chat box. So I guess the time is already used up. So
however, if there will be further questions, you can send me. I can answer by email.
Okay. All right. So thank you. So yes. So everyone.
There is raise hand question, Pak Wayan, if there is still time.
Okay. Yes. Rizky Wulan, please. Very brief.
All right. Thank you very much for the opportunity. It was interesting talk, Dr. Ernesto. So I'm a master student in marine science from Diponegoro University, Semarang. And last time when I went to go diving, I saw a lot of Joruna funebris. That's like,
great.
Yeah, and that's found a lot in a very higher sedimentation area. And why
did you find the sponge, the blue sponge?
Yeah. So do you happen to know why Joruna funebris found a lot in the highly impacted in a sedimentation area, and why Joruna is also found near the blue sponge? Thank you very much.
Yeah. No, Joruna funebris is particularly interesting because from Joruna, some years ago, I found Joruna funebris in India and in China, and some colleagues found Jorumycin, a compound in Joruna that was slightly modified by a farm company, and now is an anti-tumor compound that is used to treat cancer. So this is a very interesting topic. When we started this nudibranch, however, I did not I did not analyze the distribution of the compounds in the body of the nudibranch. I would be very interested to see if these compounds are feeding deterrent. because still we do not know. There are many information in pharmacology for the use of these compounds, but we still did not tested the compound against potential predators. So let me hope next time we can organize some assays with Wayan. If you have this easy access to your Runa funebris and the sponge, this would be great.
Okay. All right.
Thank you so much.
Thank you. Rizky Wulan, I think it's a good opportunity for future collaboration, so we can do a feeding deterrence assay with the nudibranchs and also the sponge if you would like to do it in Indonesia. Okay, thank you. So if you still have any question or something to say to Ernesto, just send a quick email.
Can I say another thing? Please,
yes, sure.
It will be very interesting to study, to understand, given that the compound most likely come from the sponge, or maybe it is slightly modified from the sponge by the movement. It will be very interesting to see if some symbiotic bacteria are involved in this biosynthesis.
Okay.
Yeah.
Okay.
In the sponge.
Yes. Always bacteria. Yep.
In the sponge.
All right. All right. Good. Good. Thank you. Okay. So I think the presentation and discussion from Ernesto is already finished. We don't have enough time to continue, unfortunately, but the rest of the question, please just send a quick email to Ernesto. Okay, Ernesto,
thank you.
Please stay with us until at the end of the talk. Now, next we have
thank you.
Another
thank
speaker.
you very much.
Bye. Thank you.
Yes, thank you. Bye. We have now Professor Ocky. So you have 40 minutes for your talk, and then if you are already ready with your slides, please just do it now. Time is yours.
Let me share first.
Okay.
Could you see my presentation?
Yes, is already on the screen. It's perfect.
My talk is about marine microbial symbiont, and currently I'm work for the Research Organization for Earth Sciences and Maritime at Indonesia National Research and Innovation Agency. Pak Wayan, my question, do I need to speak in English or can I speak in Indonesia?
Bilingual, Prof.
Okay. Okay, but first, as we have understood that marine biodiversity hotspot is representing the very diverse biodiversity in but also representing a threatened ecosystem. We are at Indonesia is quite lucky to have a very productive and diverse coastal ecosystem, namely coral reef, which represent about 14% of our world's coral reef. And then the mangrove, that represent about 20-30% of the world's, and also seagrasses. But I don't have a percentage of the seagrasses to the world seagrasses volume. As you can see here, we have very diverse in terms of marine biodiversity. As you can see here, we have seagrasses about 16 species, and then reef fish, stony coral, microalgae, sponges, 850 species, mollusks, about 2,500, and then crustacea, echinoderm, etc. And it is very unfortunate from a conservation point of view that many of these diversity produce a lot of secondary metabolite. But the problem is in terms of supply, because to get enough extract for the pre and clinical trial, we will need a lot of volume. And unluckily, those compounds are not produced in high amount in terms of diversity of these animals. Now I will come to the correlation between chemistry and biology. In the chemistry, it is represented by natural product chemistry, and for the biology, represented by chemical ecology. In this case, is marine chemical ecology. Chemical ecology and natural product chemistry are linked in a productive partnership, aimed at clarifying and the chemical basis of ecological and behavioral interaction in the nature. So before natural product chemists can explore further about the potential of secondary metabolite for industry, medicine, and agriculture, it is necessary for them to have a basic knowledge on marine chemical ecology. And I believe Ernesto has already mentioned in detail about the chemical ecology. So it is very important, and it is very unfortunate that the field of marine chemical ecology is not mature yet in Indonesia. Only limited faculty in Indonesia that provided marine chemical ecology courses, including in Diponegoro University. Here are some of the identified roles of secondary metabolite produced by marine animals and plants. For example, we have identified that some of the secondary metabolites serve as antifouling agent. For example, here in the case of a soft coral, you will never found any fouling organism that occupy the surfaces of soft coral. I think Ernesto also mentioned about the anti-predatory activities or potential of secondary metabolite, both in the toxic level and the non-toxic level, such as unpalatability. The compound of the secondary metabolite also has a role for space competition among the animals in the coral reef. Also, it will enhance the reproduction rate by protecting, for example, the larvae of the animals in a coral reef by producing secondary metabolite that will protect them from the predatory. Just like us, many animals or invertebrate could be the target of microbial attack. So for that, they have to maintain a mechanism to protect them from microbial attack. Just like most of the women in the world, marine invertebrate also very sensitive for the UV radiation. So this animal also develop and produce a compound that serves as a sun protector. Also, they will produce or make use the secondary metabolite for the communication among their species or among intraspecies, and it is mentioned already by Ernesto. Last but not least is that secondary metabolite also serve in the host and microbe interaction. That means the host of animal or invertebrate, they have used the secondary metabolite to select and to have the most appropriate microbe that will live together with the host. When we talk about biological diversity, that means we are talking about the chemical diversity. When we or you can maintain the diversity of particular habitats in the marine environment, for example, in the coral reef, then basically you will also maintain the chemical diversity. That means you will be able to find good biological diversity ecosystem. Now we are talking about marine biodiversity, marine bioprospecting. It is the scientific search and exploration of marine indigenous genetic and biological resources for commercial exploitation in order to produce commercial or industrial product. So we can categorize different marine biological resources, for example, fungi, then actinomycetes, and then mollusks, seaweeds, and other invertebrate, such as sponges. It also including trading of indigenous genetic and biological resources in order to develop and produce products such as enzyme, drug, perfume, extract, colorant, and other. So basically, when we are talking about bioprospecting, is to bring biodiversity into economic value. So in this regard, we will refer to our Convention on Biological Diversity and also Nagoya Protocol. Now, how we can make use our diversity to chemical diversity for the benefit for human and ecosystem health. So basically, we are talking about marine natural compound. Then we can divide them into three different aspects or perspective. Number one is the biodiversity. But the biodiversity, we are talking about different marine habitat will support different biodiversity, and we will have different microbes from different habitats. And then it is necessary for us also to have a phylogenetic relationship among the animal or microbe that are inhabiting a certain habitats. And secondly is the chemodiversity and biological function. So when we are talking about the functional biodiversity, that's been how in particular condition the component of diversity will act in certain way that will, for example, can be used for discovering new antibiotics. As you can see here, we have bioactive isolate that are very active against particular diseases. And then it is very important for us to have the metabolic profiles, because in many cases, when you conduct a study or extract some sample, that at the end you will have a similar metabolic profile. That's mean that will not represent a new compound. And also it is very important to study about the microbe and microbe host interaction. And last but not least, we are talking about the biotechnology and application. For example, how our study on molecular biology by studying genomic can also influence our biotechnological production, and also some patented compounds such as antibiotic, anti-cancer, antioxidant, etc. This is an identified Indonesia marine biodiversity potential. It is reported by Hanif et al. 2019 in Marine Drugs. As you can see here, we are quite happy with our diversity. And then for the bioactivity potential, you can see here we have cytotoxic, anti-cancer, and then neurological, anti-tumor, anti-diabetic, ichthyology, etc., etc. So from this table, you can see how diverse the function of secondary metabolite produced by animals and plants from Indonesian waters. Munro et al. In 1999, he proposed that mean you will be successful in obtaining secondary metabolite producing organism. So the formula is to obtain secondary metabolite producing organism, you will have a sample, time, diversity, and time assay. The more bigger and a lot of sample you collected, that means you will have more opportunity to have the hit species. But the problem is that many samples from marine environment are very slow growing species. That means you cannot collect a lot of sample in order to make some screening to have the hit species. And the second one is diversity. This is why it is very important to maintain our marine biodiversity. Diversity means across phyla. The more you have different kind of phylum, that means you will be able to have more opportunity to have the hit species. So in this case, when you have a limited budget and time, it is good to have a different phylum in terms of collecting the sample. Instead of collecting only sponges but different species, it is recommended to better collected sponges, tunicate, soft coral, etc. in the same time from the same site collection. And number 3 is assay. That means if you performing screening against fungal pathogenic, out of 20 sample, for example, only one show activity. It doesn't mean that the remaining 19 sample are not active. Maybe if you have another assay, for example, anti-cancer, out of 19 inactive sample, you may be able to find 5 different anti-cancer activity. That is the most important concept or formula of hit species proposed by Munro in 1999. I heard one of you ask about the problem of supply and sustainable use of our marine biodiversity. This is an example of 3 different compounds that is very extreme in terms of obtaining the pure compound. For example, anti-cancer ectenacidin 743 produced by tunicate Ectenasidia turbinata. To obtain 1 gram of the pure compound, according to Mendola 20, you have to collect 1 metric ton biomass or wet biomass of the tunicate. So you can imagine, just to obtain 1 gram, you have to crush about 1 metric ton. In the case of helicondrin produced by Spong Lysodendoric, even more extreme. To obtain 300 milligram of helicondrin, you have to extract about 1 metric ton of sponge Lysodendoric. And it is estimated that when helicondrin enter the market, the need for the market is about 1 kilogram per year. So you can imagine how bad this extraction or collection or sample which will have a negative impact on the marine environment. Also, in the case of anti-inflammatory pseudotorepsin produced by soft coral, it is also you have to collect and crush about. So, in this regard, we have to think about the possible or alternative to obtain sustainable sources of active compound. So this is the supply problem in initial step of drug discovery. So, for example, in the context of replicability, the loss of sources, to have clinical trial, and then commercialization. Of course, the best way is to have chemical synthesis, but not all of marine compound are viable for or feasible for chemical synthesis. Why? Because they have a very complex structure that making the cost of chemical synthesis become not feasible. Second alternative would be mariculture, both in the in situ or ex situ control environment. There are several efforts have been conducted in order to obtain bioactive compounds through aquaculture. But the problem is that the production of secondary metabolite is the reflection of the ecological pressure. For example, you will be able to grow them in their original depth, for example, 20 meters, and then you culture them at 10 meters depth. The animals or the animal producing compound, they may grow very well at 10 meters depth, but the compound you are looking for is not produced. And it's happened in many habitats. So it is very important to have a deep and thorough understanding on the in situ and ex situ aquaculture. The last one is microbial assemblage, or you can make it as a microbial symbiont, etc. It is an example of sustainable fermentation, and there is no need to do the recollection, but we have a problem in terms of unculturable microbe. As you may know that at the moment with the existing methodology and media, only 1% of the marine microbe are successfully isolated, and the remaining 99% are still out there. That means we need another complementary techniques to in order to make maximum effort of these sources, especially the marine microbe. Now we are familiar with a term called HMA invertebrate. That means high microbial abundant invertebrates. This is an example how chemical interaction between marine organisms as a host with their associated microbe or symbiont. On the upper picture, you can see how the blue sponges is contain a lot of or high microbial abundance. It refer like as city of microbe. On the below part, you can see sponges from Mediterranean also show a very thick and complex microbial symbiont. So by doing this, by performing study on this microbial symbiont, it opened an opportunity for us to explore about the potential of marine microbial symbiont as a source of sustainable secondary metabolites. This is an example of two different evidences that confirming marine natural product. That means this microbial symbiont have a very important role in the synthesis of the secondary metabolite by the host. For example, in terms of bryozoan Bucculanaretina, Bucculanaretina will produce an anti-cancer bryostatin A. But when the larvae of Bucculanaretina in the aquarium are given antibiotics that will kill the bacterial symbiont, only one bacterium that associated with this Bucculanaretina, and unfortunately they are unculturable. That means so far we cannot culture them. So by giving the antibiotic, the bacteria will die or statin A. So there is a dependence of the Bucculanaretina to their microbial symbiont. In the context of tunicate, for example, Lysoclinum patella, it has a Proroclone sp symbiont. And when we remove this symbiont, then this tunicate will fail to produce patelamide A symbiont product. By studying this phenomena, that will make us quite sure with the possibility of the role of marine microbial symbiont as the source of sustainable bioactive compound. Now, for example, I will say this is one of the secondary metabolites that serve quite broad application in the industry. This is called biopigment. So biopigment can be used as a natural food colorant, healthy food. You can take it for your health, and also biocosmetic, antioxidant and anti-aging, pro-vitamin A. Also it's used for photodynamic therapy for cancer. And then it is also prospective for pharmaceutical industry. It can also use for biohybrid solar cell as well as biofuel, biodiesel, etc. This is an example from our study in our group in Marine Tropical Biotechnology Laboratory in Undip. From red Gorgonia leptogorgia species, the red one, we was able to isolate the red pigment producing bacterial symbiont. And from soft coral Sinularia species, we was able also to isolate the yellow pigment producing microbe. So this again will show that there is a quite strong relationship between the host and the symbiont. When we talk, for example, about carotenoid-producing bacterial symbiont, pigmented marine bacteria vary significantly from 10 to 70% of total colony-forming unit, comprising primarily of the color of red to yellow. In most cases, based on our experience, you need to use a rich medium in order to obtain quite high percentage of the pigment producing microbial symbiont. This is our result that has been published in international scientific journal. Pigment from bacterial symbiont of soft coral Sarcopithon from North Java Sea. As you can see here, we have quite happy with the result of our ability to isolate the pigment producing symbiont. And quite nicely, So, for example, the isolate CBSTP11 quite similar in terms of antioxidant activity with the standard of beta-carotene. In another work, we also even found isolate that producing carotenoid with beta-carotene activity or antioxidant better than beta-carotene. So I believe still there is so much possibility for us to isolate and to make use this potential for our particular purposes. This is an example how the importance of alpha amylase or application of enzyme alpha amylase, detergent, bakery, and textile. Annually, this quite high market of business of enzyme in the world. So it is important for us to always screen new possible sources of alpha amylase. And this is an example enzyme produced by symbiont of soft coral with application in industry. So with the screening method, as you see in the plate, there is a clear zone around the media of bacterium Bacillus aquamaris that produce enzyme with the ability to hydrolyze different starch granule. As you can see here in the right side, they are able to hydrolyze almost so diverse different granule of starch, for example, corn, cassava, sagu, potato, and rice. So based on our study in different sampling site, we were able also to have a new type or new strain of enzyme, alpha amylase enzyme producing microbe. This is my student working on the prevalence of clinical isolate, especially E. coli multi-drug resistant extended spectrum beta-lactamase. So it's quite a high number in our region in Indonesia, up to 25% cases of E. coli MDR ESBL. When you have a pathogen become multi-drug resistant, that means they will resistant into the available commercially antibiotic, and this will become a problem because to have a new antibiotic, it takes between 8 to 12 years before it come into the market. So one of the potential of microbial symbiont with tunicate, here we have Pseudoaltromonas rubra TKJT 2010, and we have, using our screening test, we will be able to obtain a pure compound that have anti-MDR activity against Pseudoaltromonas, and the pure compound is identified as isatin. So by giving you only several examples of the role or functional diversity of marine microbial symbiont, it is still open for you to further explore the richness of marine microbial symbiont from different habitat and different hosts. For example, tunicate or soft coral or mollusk or nudibranch, etc., and different unique habitat that can be used for further exploration. And as I mentioned in the previous slide, that only 1% of marine microbe has been successfully cultivated. So we need another complementary technique, which we call metagenomic, by direct isolation of DNA from environment for DNA knowledge application. And also, this is also an example how we are challenged by the presence of a particular spawn, for example, Theonella swinhoi. This is very quite distributed spawns, including in Indonesia. And it is known that they produce so many bioactive compounds with different activities, onamide, auranthoside, and then nasumamide, orbiculamide, pseudonalamide. You can imagine if we insisted to collect all Theonella swinhoi to produce this active compound, you can imagine just within 1 year, these sponges will be extinct from Indonesian water. But this Theonella swinhoi is part of the high microbial abundance invertebrate. As you can see here, we can see the spawn bacteria that exist within Theonella swinhoi. So 40% of the biomass consists of bacteria. But unfortunately, more than 20 species, almost of them cannot be cultured. You cannot culture them in the laboratory. So for that reason, you need to do the metagenomic study. And this is an example of marine metagenomic portal. This marine metagenomic portal was made online in 2017. It is maintained by the Center for Bioinformatics at UIT, the Arctic University of Norway. So if you can see here, compared to the left side, there are so many blue dots. That means there are so many studies about metagenomic in that area. But when you see at Indonesian area, there is no single dot representing the sequence of the marine metagenomic from Indonesia. The use of metagenomic is quite increasingly active, and it is used in different range of scientific and industrial application. For example, you can see here from monitoring pollution and then recycling water system to disease surveillance and develop functional feed for aquaculture. Now, with the development of biological molecular, especially molecular biology in terms of metagenomic and the new development of synthetic biology approach. So in this regard, basically you can combine the metagenomic approach with synthetic biology. From metagenomic approach, you will be able to have functional metagenomic data analysis, different sequences with different potential for the synthesis of particle bioactive compound. On the other hand, with synthetic biology, we are talking about biopart, coding area of gene of interest or other region of the gene. For example, here you can see that interchangeable biopart, that means we can isolate biopart that have different serve function, for example, as promoter, gene of interest, terminator, and ribosome that is set in a certain size. So it can be interchangeable so that it can be used for us to build up genetic circuit. And by doing so many different circuits, so basically we will be able to have high value compound from our metagenomic study. And synthetic biology can be performed by DBTL cycle. That means we can do the design and then build, test, and learn. By doing this, we will be able to obtain the best assembly through combinatorial assembly of different bioparts. For example, in the pathway 1, you will have different biopart circuit compared with the pathway B that representing different circuit. So by conducting this repeatedly, so you will be able to develop new product based on metagenomic and biosynthetic biology. To conclude my remarks, much of nature treasure trove of small molecule remains to be explored, particularly from marine microbial symbiont to fulfill the human need. Thank you very much for your kind attention.
Wow. Okay. Luar biasa. Fantastic. So thank you very much, Prof. Ocky. I do agree with your closing statement. So there are the current research updates from Professor Ocky, and also he highlighted some inherited challenge in natural products research, that is the supply problems. But Prof. Ocky already mentioned some alternatives and possible future development, how to tackle these supply problems. Okay, from Ocky, in the chat box, we have already got 2 questions. So I will read it out for you. So the first question is from Dwi Imam. Okay, I'll read it out to Mr. Ocky. I have preliminary for extraction holoturin substance from sea cucumber with green solvent. This method allows me to harvest substances without kill the subjects. So he actually have an experience in using green solvents, and then perhaps could be good information to tackle the supply problem. I think something like that, the questions. Well, so can you make comments about this?
Yeah, thank you very much. I know Pak Imam, and I think he's doing Ph.D. on that marine natural product, and I'm extremely very glad to hear his advancement of the technique he used without harmful to the marine environment. So yeah, of course, as long as I think one of the technique several years ago, for example, to collect the bioactive compound from the field by covering the sponges, and then they do the squeeze to obtain different extract also. But what Pak Imam mentioned about this green extraction, I really appreciate and would like to hear more from him next time. Thank you.
Okay. All right. So you know already him, and then the green solvents. Yes, it's from the names. It's just promising for future usage of this solvent. Okay. Then the next question is from Siti Nurul Aini. I guess she's now at the last year of her PhD. She's in Okinawa. So hello from Ocky. I am very happy to listen your lecture again after more than 10 years. So I guess she's your former students. So hope you are always healthy. Okay, I'm interested in the micro symbionts in sponges and corals. Is that possible? The same micro symbiont in different hosts has different potential. So yes.
Based on our experience so far, there is what we call it a general microbial symbiont. That means we can find them in quite wide range of hosts in the marine environment. Some of them, we call it species-associated symbiont. That means certain symbiont will only associated with certain hosts of animal or plant in the marine environment. Of course, as I mentioned, this is one of the role of secondary metabolite make use by the host to select the most appropriate symbiont. It is quite interesting for to know that one of my students several years back, he studied and compared the symbiont from seagrasses, and then she also isolated the microbe from the surrounding water.
Okay.
It is quite interesting that when we do the antibacterial activity, the symbiont, they did not inhibit the growth of the other symbiont, but they did actively inhibit the growth of the bacteria isolated from the surrounding. So based on this experience, I think there is a certain role of secondary metabolite in terms of selecting and stimulating the formation of biofilm in the surfaces of particular host. And of course, even from the same species of the host animal of invertebrate, but they come from different activities. They may have different potential in terms of the biological activity.
Okay.
One example is conducted by my ex-student. She collected the sample from North Java Sea, from the sponges from Java Sea, and directly isolated and getting the extract. And then on the other hand, she grew the sponges in the aquarium for the 1 month. It is nice to see that based on the extract profile, we can see that even though they are the same species, but they grow them in different habitat, they produce different secondary that the biological activity is the reflection of the ecological pressure in certain habitats. So that is why they will produce different compound. Maybe in one location or habitat, the ecological pressure is the microbial infection. On the other hand, on different location, the ecological pressure may be predatorism. So different compound will be produced in order to protect them from different threat that they are facing. That is why it is very common to have different biological activity, even though from the same species, but they grow in different habitat or site location. Thank you.
Okay, thank you, Prof. Ocky. So yeah, so ecosystem, the surrounding area are greatly influence the chemistry of the symbionts. Okay, in the chat room, they're already empty, so there's no more question, but I guess Ernesto wants to ask question. Okay, Ernesto, yes, please ask your question to Prof. Ocky now.
Thank you so much. It was a really great presentation. Thank you. I have a problem with some gorgonian in the Mediterranean. I would like to hear your opinion about this. Recently, we found that these gorgonian, which are red, they can be red and yellow. This is the same species. However, independently from the color, we found that they can be different by chemotype. They can contain one compound or another compound, independently from the color. So we are working on this. We are trying to understand this. However, about the color, very recently, last month, my colleague Carlo Serrano, I don't know if you know him because he was several times in Sulawesi, in Indonesia, but Carlo Serrano. So he found that this gorgonian can change color. However, we maintained for 1 year this different gorgonian with different color in aquarium, and nothing changed in the chemical composition. Now, I suppose now something occurred in the maybe there are some bacteria producing these pigments, this color. I don't know how we can approach this problem. Thank you. If you have some suggestions.
Okay.
Thank you, Ernesto, and thank you for following my talk. I thought you have already left for different activity. I think there is one what we call it hypothesis, that is probiotic hypothesis. That means many marine invertebrate, especially in the coral reef, they are able to maintain dynamically their symbiont in order to cope with the ecological threat. So in certain time, not only in gorgonian, even in a certain coral, we can see that they are keep changing the color due to the different composition or holobiont that residing within the coral or gorgonian. So I would suggest that you do the combination of genome and metagenomic to see how different the composition of microbe in certain period when you see different color. And I'm quite sure that it will be as a result of the dynamic of different holobiont that are associated the microbe. So on the other hand, as you mentioned that you maintain and grow them in the aquarium and they are keep in the same color. So that could be that ecological threat that they are facing quite different when you found them in the natural environment. So yes, I cannot say anymore, but I believe that this will be an example how you may relate the ecological pressure with the changes of the color of red gorgonia. Thank you very much.
Okay.
Thank you so much.
Yeah.
Okay.
Thank you.
All right. So now we are at 4:41 p.m. So I guess there are no more questions on the chat room. So I think my job as moderator is almost finished. Okay, Resi, can we close the presentation and discussion session?
Yes, please.
Yes.
All right, fantastic. Okay, everyone. Dr. Ernesto, Prof. Ocky, and Pak Gunaja, and all the participants. So we have been following the discussion and the presentation from two speakers who are really knowledgeable in their respective field of research. And there are also plenty opportunities for us to do future research collaboration. So this webinar, this seminar would be as an entrance for all of us. So if you need contact details of Professor Ocky and Ernesto and asking about their talk and everything, then please just do it by email and contacting us if you don't have the email address of them. So we are opening actually for the research collaborations for doing research in this challenging area. Okay, so as a moderator today, I would like to thanks again for the speakers. And then before we leave, then please let me share a certificate of appreciation to both of you. Dilarisma, please share the certificates. Okay, this certificate from MERO Foundation is presented to Dr. Ernesto Mollo as a keynote speakers in this webinar series. Okay, so the certificate will be emailed to you. Next, another certificate is also presented to Professor Ocky Karna Radjasa as speakers. Okay, thank you so much for coming today.
Thank you very much.
Yes. Okay.
Thank you,
You're
Ernesto.
welcome. Okay.
Thank you, Ocky.
Yeah.
Okay. Okay. Thank you so much for everyone who attending the seminars today. We are looking forward to welcoming you again in the next seminar series. So if you have any questions regarding MERO Foundation, what we are doing and what we are offering, please don't hesitate to join us and contacting us at our social media and our website. Okay. Thank you, everyone. Thank you.
Ciao.
Have a nice day. Ciao, Ernesto. Thank you. See you again sometimes in Bali.
I hope.
Good afternoon, everyone.
Thank you.
Prof. Ocky, matur suwun, sukma. Terima kasih
Satu.
banyak. Okay. All right. Resi, please,
Bye-bye.
it's your time. Bye-bye. Ciao.
Bye-bye.
Ciao, ciao, Ernesto.
Yes, thank you, Pak Wayan and Ernesto.
Okay.
Yes, Pak Wayan. Maybe if Ernesto still have time, we can have a little.
No, I can. Okay.
Session.
I'm here.
Yes, yes. Please, Resi.
Great.
Yes. Yeah. Thank you, Ernesto, for your very nice talk.
Your camera, Resi.
You're welcome.
Oh yeah, sorry. Okay. Yeah, Prof. Yeah, Dr. Ernesto. Currently, we are working on nudibranchs in MERO Foundation.
Yes.
So in the future, maybe we would like to more discussion about how to develop the experimental section in the lab. Because we do not have any more idea on that. So currently, for me, we are in MERO Foundation. We are working to know how about the chemical interaction in the ecological in the sea, and also in the microbial. So I'm doing the isolation of the bacteria from the nudibranch between the food preference. So from sponges, from hydroid, and also algae, because each of them has different food preference. So I would like to know how far the chemical cool between them and the bacteria isolation. So we try to isolate the bacteria from the algae and nudibranch, but based on the specific food. So we collect them when they are exactly feeding on it. And we have the picture. So, and then we isolate them. So we would like to have an idea about the chemical and then the experiment in the lab, and also the Joruna funebris that previous question from Mbak
Yes.
So
this is extremely interesting.
Yeah, we really found to date in Jepara actually. There are so many Joruna funebris also with their sponges, the blue sponges. Yeah, extremely like family of Joruna funebris. They are like family. So it's more than 50, maybe 50 species together. Yeah, so this is interesting to see the chemical. Yeah, maybe
So,
in the future we would like to know how to
sorry,
design.
can I ask, did you find any time the rubescens?
Joruna rubescens?
Because it contains very interesting compounds, but I found just a few individuals in China some years ago, but still there is some work to do on, because there is a molecule with a new skeleton, and Wayan, you know, Mariana is always asking for some more material to define this structure. Yeah. This is just please let me know if you also can find your own rubescens there.
Yeah. Risma, did we find Joruna rubescens in the sampling area?
Are you asking?
I'm asking our team,
okay,
because we did survey.
We found the Joruna rubescens, but not in the sampling area. The way when we go back from the area to the villa.
Oh yeah. So we still found the specimen.
Mm-hmm. However, this is interesting. Yes, also to have a look to the other species belonging to this genus. Very interesting. Yes, okay. I would like to invite you to make some experiments with the Joruna funebris, because if you to make some food choice experiments to see if the animal is able to follow the tracks. You can okay, it's difficult. I can try to write an experimental design to do these experiments, and yes, I can send you this idea. We can discuss about this. I think it is because you know these animals can follow tracks of lipophilic compounds that are present on the substrate to find their prey. Let me see if we are able to find what is the chemical cue that attract Joruna on the sponge.
Okay. Yes, Ernesto. So yeah, so maybe this is just beginning of our issue. So next time we can have another email and Zoom meeting, maybe Ernesto, to design some experiment.
I can send you some previous paper on this topic to see, because however always these experiments were biased, because always people, when also my colleagues from Spain always tried to use the compounds vehiculated with the DMSO, vehiculated in the water, but this is not natural, you know, huh?
Yes.
And,
yes.
Because these animals do not swim, they do not follow in the water. They track the signals on the substrate. Okay. So let me see. This is a new field.
Yes. So which means that this seminar open up new opportunities for all of us. In MERO Foundation, we have plenty of young scientists, beautiful girls there in the lab. And then also they are doing diving, scientific diving every week. So so we are waiting for volunteers as well now. So if you have students in ICB in Italy who wants to do volunteers work doing experiment in Bali, then we are most welcome to do that.
Okay. Now I have one PhD student. She's Carmela Celentano. And she is a diving instructor.
Ah, lovely. Okay.
So,
all right.
She's so now here with me.
Yes.
It will be the right person.
It's a good match. It's a good match. So yeah, we need to follow up this meeting by another meeting again. And then let me set up, let we arrange for the time and then what we are going to talk. At the end of the day, we are going to find a research grant that will allow us to exchange scientists, maybe some of us from MERO going to Italy to learn the
of course,
or maybe you and your colleagues are coming back to MERO Foundation and stay in our place for a certain time to do the experiments.
I noticed that in the program, in the MERO webinars, you also organized a talk of Angel Valdez from California. Angel Valdez.
Yes.
Angel.
Yeah, Angel Valdez. Angel Valdez.
Angel Valdez. He's my good friend.
Yes.
Okay.
Currently, we went diving in Mexico, in Egypt together.
Yes.
I know him very well.
Currently, Angel also already visit, but that time there is no MERO Foundation.
Yeah. Let me hope one day to meet him again. Why not in Bali? It would be great.
Yes. And you can see Cristiano again to talk about Nudi branch in the dining room and talking until late night. Okay, that's gonna be good. Okay, all right.
Okay, thank you very much, Ernesto.
Thank you so much to you.
Yeah.
Great.
See you again next time.
Congratulations for the foundation.
Ciao, Mariana and other friends. See you again sometimes in Bali, Mariana. Okay, everyone.
They are in the lab.
Okay, all right. Just say hello to them. Say hello to them. Okay, I miss you all. Okay, bye-bye. Bye-bye. Ciao. All right.
Ciao.
Okay. Gimana? Sudah diakhiri ya?
Iya Pak Wayan.
Oke deh.
Ini kita sudah di penghujung acara, jadi bisa live pesertanya. Ini saya Pak Wayan kalau sudah selesai boleh ini. Tapi ini saya mau menjawab ada yang bertanya Jeremy tentang MERO Foundation, jadi nanti bisa saya jawab Pak Wayan.
Okay. All right. So have a nice day, everyone. Take a rest and then thank you all. Bye bye.
Yeah. Thank you very much Pak Wayan. Oke, untuk Jeremy tadi ada yang chat ya. Oh, ini ada banyak sekali chatnya. Jeremy, silahkan ditanyakan kalau ada yang ditanyakan tentang MERO Foundation. Sebelum kita tutup. Bisa open mic ya.
Halo, selamat sore Kak Christina.
Iya. Untuk peserta yang lain boleh leave ya kalau misalnya sudah selesai.
Iya, baik Kak.
Iya.
Kak, barusan saya habis ada dapat, saya boleh open cam nggak Kak?
Iya, boleh silakan.
Iya Kak, ini dengan Jeremy. Jadi barusan saya habis dapat ada sertifikat dari MERO Foundation. Kalau misalnya ada nama Kakak di sana, tentu saya itu adalah Kakak sebagai chairman dari MERO Foundation. Apakah itu benar?
Iya.
Saya ingin bertanya Kak, apakah MERO Foundation ini adalah suatu organisasi yang khusus bergerak di bidang riset seperti halnya LIPI, tapi badan swasta atau bagaimana kira-kira?